How many of you may be looking at this and saying, I have no idea where to start? Okay, so we've read the problem. Now, what is the difference between a monohybrid cross and a dihybrid cross? What do you suppose di means? Two traits, like height, texture would be two traits. Or coat color and horns, that's two traits. But if it's just coat color, then that's not a dihybrid cross, but mono. That's just one contrasting trait. So I see you've done them both already and got that right. Okay? So, in other words, what we're going to do is look at this first part, then we'll put two together for a dihybrid cross. All right. So, the reason that we see this, an incomplete dominant cross, why is that important, do you think? What happens on the color wheel if you mix black and red? You don't get purple? Oh, okay. Well, if you mix black and white then you get gray. Okay? No one would argue with that. Okay? But this is an example of a red pole, kind of similar to a red pole and a Charlet. All right? The reason we say that, okay, if this was a completely dominant cross, okay, well then what we would do is, let's say capital P is purple in these petunias, okay, and then we'll say lowercase p is white. That's if this was a completely dominant cross, like plant height, pod texture in pea plants. Those are where you have dominant traits, but there's times where you have incomplete dominant crosses and dominant. So if this is an incomplete dominant cross, we know that what would this look like if we have two capital P's? It's going to be purple. What happens if we have two lowercase p's? It's going to be white. All right? But if it's an incomplete dominant cross, if you have this, okay, if it was completely dominant, this would be what? Purple. But if it's incomplete, that means it doesn't fully mask that over. What's the contrasting trait for purple and white? What does it tell you in the problem? Pink. Okay? So here we have three different genotypes and then three different phenotypes. Three genotypes, three phenotypes. Okay? Unlike if we're saying that tall is dominant over short, what does this look like if T is for tall? What about if we have two lowercase t's? Then it's short. Well, what happens if you have one uppercase and one lowercase? It's still tall. Okay? Three contrasting genotypes, but only two phenotypes that you can actually observe. I think some of us are possibly getting these two mixed up, but you have to be told that it's an incomplete dominant cross. That's why we're going over this. Who's lost? Okay? So, do we get this part here, our key? Okay? But if it's an incomplete dominant cross, it says we would get a pink, okay? A cross between, genotype of a cross between a pink and a white. What we're saying here is that when you cross these two, you get that third possible outcome, okay? Because it's an incomplete dominant cross. Just like when you see this for, we know that that's red pole for cattle, right? Because that's a lot of variables you used in your key, correct? No one would argue that. Okay? But then what does that result in? Rhone. Okay? And then this results in your Charlet. Again, this incomplete dominant cross means they're both mixing together to give you a third outcome. Okay? Now that's a little different than a codominant cross. So what would happen is if I had, let's see here, I don't have the correct colors up here, but what we're saying is if we take all this black, okay, and if we had a white marker, if that was possible, if this was paint, then you take white paint and you mix these two, what do we get? It's not black, it's not white, it's gray. Okay? So in this case, when we see a cross like this that's incomplete dominance, just like this color here, we see one that's completely purple, one that's completely white, but when you mix the two, you get a pink outcome. If you mix black paint with, use your imagination, white paint, when you mix them, you get a third outcome. You don't have black paint, you don't have white paint, you've got gray paint. Does that make sense? So that's what we're trying to illustrate when we see these types of traits. Okay? When it's incomplete dominant, you would have to be told that. Okay, so in this monohybrid cross, just one trait. Okay? I shouldn't have erased those, but I did. So we know that this is purple. We know that this is white. Then if we have one of each, we know that it's pink. Okay, so in this monohybrid cross, what happens when we cross a pink and a white petunia? Okay, so what's the genetic makeup for a pink petunia? What type of peas? Big and little. That's a heterozygous trait. Okay, so big and little times, what's the genetic makeup for a white one? 2 little peas. Okay? So since this is monohybrid, it's only one trait, so therefore there's only 2 alleles in here. Okay? So how big is your box going to be in a monohybrid cross? 4. A 2 by 2. Okay? So we can put these over here and these across the top. Okay? So once we got this, now you got the hard part done, especially in these smaller crosses. Okay? So whether you go this way or this way, it doesn't matter. I just prefer to go left or right. Okay, now looking at this, what are we referring to when we say genotype? That's what this G is for. We're talking about the genotype. It's the what? It's the genetic makeup. That's what you see inside these boxes. So therefore, you just pick a box, okay? So we got a capital P and a lowercase p. How many of them do we have? Okay, so just 2 of those. Okay, then the other 2 outcomes are what? 2 lowercase p's. Okay, and I'll just put them below here. Okay, so 2 heterozygous trait, a capital and a lowercase p, 2 lowercase. Okay. Then, once you get the genotype, that's the different combinations that you have, how's that different than the phenotype? The phenotype is what? What you actually would observe. The reason I point at our eyes is that's what you actually would see and observe. So, what that means then, the phenotype. In other words, two of these are going to be what color? Pink. Okay? Oops. Two pink. And then what's the other one? Two white. Okay? Remember, on your papers, the genotype is just the genetic makeup. The phenotype is what you actually would observe. Okay? All right. On that top problem, what's not in the box? Is anybody confused? Left outside. Left middle. Right middle. Because if you're nodding your head yes, that means we're going to keep on going. Right middle. Right middle. Okay? Right outside. And front. Okay. Fair enough. Okay. Now, the reason this is inside the box is it says, then what happens if we're throwing in a height variance in here? But the difference between, what is it saying here? Tall is dominant over short. So that means if that capital letter is present, that outcome will be what? It will be tall, even if there's only one of them. It's either a tall or a short. There is no contrasting trait like there is with an incomplete dominant cross. Okay? So, if we're just doing this little 2x2 in here, okay, again, we make a key. Capital T is tall. Lowercase t is short. Okay? So what it's saying is one plant is heterozygous for height. What's heterozygous mean? Okay? So that means we're going to have a capital T and a lowercase. Okay? Because it's heterozygous. They're two different, just like what you said. Okay? So then the other is short. The genetic makeup for that, according to our key here, is what? The two alleles are what? Is there any capital letters in there? No, because if that trait is present, it's going to be tall, because tall is dominant over short. It doesn't say codominance. It doesn't say incomplete dominance. It just says this is a dominant trait. Okay? So then, when we do this cross, again, a monohybrid cross, okay. So, the genotype for this, okay, we've got two like that. Here's our genotype. And then we've also got two lowercase. Okay. Now, if this was an incomplete dominant cross, if it was, okay, just for argument's sake, if this was incomplete dominance, well then, let's see here, this would be tall, this would be short, but if it was incomplete dominant, what would you get? A medium. You get something in between, just like what we do here. This is incomplete dominant cross, where this is something in between. But that is not the case when it comes to height, because it says that tall is dominant over short. So, our genetic makeup then says two like this, two like this. Then, what would we actually observe for our phenotype? Too tall and too short. tall. Even though, okay, even though that recessive trait is in there, the tall trait is dominant over the short. So if that tall trait is present, it's still going to be tall. Okay, now if we put the two together, okay, now if we put this whole thing together, can we close the books on this? Okay. So now if we take this whole problem as a whole, okay, what happens if we cross a pink, which our key is over there, and we have a heterozygous for plant height, so that means there's one of each, okay? So if you understand this, okay, why is there 3 different outcomes over there and only 2 over here? It's not incomplete dominance. If you understand that, you are well on your way to navigating your way through this. Now it's just a process of not going too fast and being able to read your own writing. Yeah, would probably put me to sleep too. No, that's fine. That's fine. Okay. So now that's one trait. Okay. Now, what's the outcome for a white petunia? It's genetic makeup. What type of peas? Small peas. That's why it's important to make this key. All right. And then, what's the only way this can be short? Okay. And notice, again, since this is not an incomplete dominant cross, if we had this, it's still tall. Because this is complete dominance, this is incomplete dominance. Okay. So, if you were to set this up now, this will be the last thing that we do. And we're not going to have time to work through it. But maybe something like this could appear on your problem quiz tomorrow. You just never know. So some people might be sitting there, shucks, why didn't I write this down? Is that anybody out there? Okay. It is. Okay. So now as we look at this, does anybody have a problem foiling this out? Good. Okay. Otherwise, if that's the case, what goes here then? Okay. Do we have to worry about foiling this one out? No, because they're all the same. How many of you knew that going into this? Yeah, because little p, little t, little p, little t, little p, little t, little p, little t. Again, they're all the same. There's no variance here. Okay. All right. So, I'll hand back some papers for you, and then we'll catch up with this tomorrow. We'll begin reviewing for your semester. Okay.